Hi there, my name is Steve Kirby. I'm a manager of data programming at Celerion and have a leadership role in implementation of CDISC standards. And I'm going to talk briefly today about what we've done to harmonize our data processes with the CDISC Family of Standards. Right now at Celerion, our data processes are built from collection through analysis and submission. They're built around the CDISC Family of Standards. And so more specifically, we map data is collected, C-data is collected to SDTM. We map SDTM data to Atom for analysis. And then we create data definition documents, Define.xmls, at each stage of the mapping. And we use those as a communication tool. All of our work is done in an industry-compliant environment, which gives us a backdrop of enhanced traceability. And so that brings me, at least it's certainly of interest to me, is how we got there. It's a series of small steps that I think have gotten us to a much better place than we were 5 years ago. And the industry in general as well, I think, has worked towards standardization. I think it's going to benefit everybody. Roughly 5 years ago, even 10 years ago when I started, we had an active programming group that mapped the data collected in SAS to many different structures. The data collected at that point was a very nice, easy-to-use format. It just didn't follow any global standards because, roughly speaking, they didn't exist or weren't public domain in a way that we could use them. Over that time, CDISC standards became increasingly relevant, starting with SDTM, the study data tabulation model associated with listings and the default submission. Formal documentation as well was becoming increasingly important, and the expectation of our clients is that became more and more useful, more and more user-friendly. And with the release of CDISC SDTM version 3.1.1, our programming group here and the scientists we work with, the data people we work with at the collection area, we're very confident that we finally had a standard that we could say, this is compliant SDTM data. This is something that we should really start to focus on because it could have a lot of benefits both internally and externally. And as we got into it, the demand for SDTM increased more and more and more. It was being adopted industry-wide. It was being expected as a default. And so we started to work to optimize our SDTM mapping process. And first of all, we looked at some of the core challenges. There are challenges faced by every company out there. We're doing science, we're not bankers, and the data is driven by the scientific needs. We naturally have input data variation at the margins for the substance, and we also have some input data variation at the structural level. Internally, that variation is less, but when we get data from outside clinic, outside labs, outside sources, it can vary wildly in structure. And then as well, we have some output data variation. By and large, in a CDISC environment, that's primarily driven by the substance of the data, but some clients have sequences of studies following 3.1.1 standards. Some have studies following 3.1.2 that they need to be consistent with. There are external standards that we have to work with at the client level as well. And so what we looked for is the right balance of flexibility and optimization in our mapping so we could meet a lot of people's needs, everybody's needs, and still be able to produce this kind of quality product quickly. And so the approach that we've taken over the past few years is to integrate metadata helper files. We use those initially to establish a conforming format to make sure our data complied at the variable names were right, the variable labels were right. And as well, for the substance of the data, the most critical issue, we built in substantive review checks into our mapping code, both for self and peer review, so we can check at every step along the way whether everything's going as expected. Once we had digested that, once we had a cleanly operating process driven by metadata for SDTM, we started to look at what can we do to minimize the input data variation at the structural level. And at that point, the CDISC standards, the data standards from CDISC associated with data collection, were becoming more and more well-formed, and we decided to move towards implementing those at the data collection level. We updated the data format to CDISC over a series of steps. We had to do that very, very carefully because the original collection data impacts everything downstream of it. That includes our data mappers, our statistical SAS programmers, and our scientists and our clients in a lot of cases. So we had to work very, very closely with all of those groups to make sure we were communicating effectively and meeting all their needs. We formed working group initially, and that was expanded to sort of a number of subgroups to coordinate CDISC and SDTM with all the people who worked with it downstream. And once the CDISC implementation at the collection level was complete, working smoothly, we started to look at what do we need to do next. And we started to look more and more at the ATOM model for analysis data. Every scientist in the world, every scientist I've ever met wants data that is optimized for their use. They want to click on a data set and have exactly what they need there. And ATOM data, with the release of 2.1, did just that. It gave us a open source, durable structure that is optimized very, very nicely for scientific use. It gives a really nice mix of flexibility and standardization. And so we worked in tandem with our scientists here and scientists externally that we were delivering data to make sure that we were doing what they needed. And as we went through that process of coordinating the data needs with internal and external scientists, with all the way downstream to reviewers at the FDA, it really emphasized how important it was to communicate cleanly about data, to make it easy for people to see exactly what the data was trying to say. And so we tried a variety of means to communicate effectively about it, but in the end we said, well, we're already, in general, as a last step, we were creating Define documentation to help explain the data as part of our submission packages. And all we could think of is if we had that available at earlier steps, what a wonderful communication tool that would be. And so maybe a word about Define.xml. It's a data definition document in XML format, which means it's navigable. It has links you can click on and look at data structure, data details, derived information. We'll have an associated comment. You can click to external links such as the CRF and quickly find out what you want to know about the data. And so we thought, why not build that in earlier in the process and use that as our communication tool? And so we had to work a little while to find a very clean way to integrate it. We had to think about it a while. And so the answer turned out to be fairly straightforward. We enhanced the scope of the metadata helper files that we were already using to drive the mapping process to include all information needed for the Define.xml. That is, all information that we couldn't derive directly from the data itself. And then we've, as planned, made Define content available as a communication tool at each stage of the mapping process and use it at the test transfer, at the draft transfer stage, as a way to communicate about data needs with internal and external scientists. And so all the way through this process over the last 5 years, we've come to the clear realization that the CDISC standards do work best when applied across the board when used from collection all the way through analysis. They work well together, and it ends up with a clean process that's where traceability can be gracefully established for reviewers and other end users. But as we're looking through this, again, we're doing science. We're not working with data that's completely stable because we're looking to answer specific scientific questions that vary with each drug compound, with each study. Inputs and outputs are variable. You have to keep processes flexible so you can meet the scientific needs of the study. And you can never communicate enough. You can never do enough outreach. And putting a lot of thought into how to do it efficiently and effectively is a very big deal, and something that we're really happy we focused on for the past few years. And I want to be clear that although I'm a leader in the area, I'm just a small part of our effort to work through integrating CDISC standards from CDISC through ADAM at Celerion. I want to thank Celerion for supporting this presentation, which was initially given at JSN, and to supporting the programming initiatives that made it possible. It takes a very big time commitment in personnel and money to make this happen, and I'm happy that they saw that it was a good thing and supported it. And I want to give specific thanks to some of the other leaders in the area. Elizabeth Reinbold, our lead programmer, Matt Weedle, one of our senior programmers who's been focused on Define documentation, Luke Reinbold, who's been working with Matt and was one of the principal architects of our Define programs as well, Alexandra Stein, who's did a lot of core work on our data definition and our input metadata for the mapping process, Carl Miller, who optimizes processes for our preferred vendors, Deidre Kreipels, the leader for CDISC, Tamara Cuddy, the Associate Director of Data Programming, and Chun Feng, who helped us move to the 3.1.2 standards from 3.1.1. And with that, I want to say if you have any questions about what data services we offer or any other details, please contact info@Celerion, and I'm sure you'll be directed to one of our subject matter experts. Thanks a lot for listening. Bye-bye.